Gibbs done in equilibrium. ChatGPT said, Gibbs done in equilibrium. Introduction. Gibbs done in equilibrium, or done in equilibrium, describes the distribution of charged ions between two solutions separated by a semi-permeable membrane, where one side contains an impermeable charged species, such as a protein or macromolecule. This concept is fundamental in cell biology, physiology, and biophysics, especially in understanding ion transport across cell membranes, osmotic balance, and electrochemical gradients. Concept of Gibbs done in equilibrium. When two solutions are separated by a membrane that allows small ions to pass but restricts large, charged molecules, e.g., proteins or macromolecules, an unequal distribution of diffusible ions occurs. This leads to electrochemical imbalance. The movement of small ions generates an electric potential. Osmotic effects. Water moves to balance the osmotic pressure, affecting cell volume. Ion concentration differences. The impermeable ions cause an asymmetric distribution of permeable ions. This phenomenon plays a crucial role in nerve signaling, kidney function, and intracellular fluid balance. Mathematical representation. If an impermeable negatively charged macromolecule, such as a protein, is present in one compartment, it attracts cations and repels anions. The done in equilibrium is mathematically expressed as C1 plus times C1 minus equals C2 plus times C2 minus C1 plus times C1 minus equals C2 plus times C2 minus, where C1 plus C1 plus and C1 minus C1 minus equals concentrations of cations and anions on one side of the membrane C2 plus C2 plus and C2 minus C2 minus equals concentrations of cations and anions on the other side. This equation states that the product of ion concentrations on each side remains equal at equilibrium. Done in potential E. DED. The electrical potential difference E. DED. Created across the membrane due to ion imbalance is given by the Nernst equation. ED equals RT ZF lane C1 plus C2 plus ED equals ZF RT. C2 plus C1 plus. Where R equals universal gas constant 8.314 J per mole KT. T equals absolute temperature KC. C equals charge of the ion F. F equals Faraday's constant 96,485 C per mole C. 1 plus C1 plus and C2 plus C2 plus equals cation concentrations on both sides. Biological significance 1. Cell membrane and ion balance. The Donen effect influences ion distribution across cell membranes, especially in neurons and red blood cells. Cells maintain ion gradients using active transport, e.g., not K-pump, to prevent excessive swelling due to osmotic pressure. 2. Blood plasma and interstitial fluid. Plasma proteins, which are negatively charged, attract non-K ions, affecting the osmotic pressure of blood. This is critical in kidney function and maintaining blood pressure. 3. pH regulation in tissues. The Donen effect influences proton H concentration, affecting intracellular and extracellular pH balance. Important in acid-base homeostasis in the body. Applications of Gibbs Donen equilibrium. 1. Physiology and medicine. Kidney function. The equilibrium influences ion exchange during urine formation. Neural activity. Ion gradients across neurons help generate action potentials. Plasma protein distribution. Maintains blood osmotic pressure, preventing excessive water loss from capillaries. 2. Biophysical and chemical applications. Electrodialysis. Used in water purification and desalination. Colloid chemistry. Determines behavior of charged colloidal particles in solutions. Membrane transport studies. Helps in understanding passive diffusion of ions in artificial membranes. Solved previous year question. Question. A semi-permeable membrane separates two solutions. One contains an impermeable negatively charged protein and NaCl. The other contains only NaCl. If Na concentration is 100 m inside the compartment with protein and 150 m outside, calculate the Donen potential at 37 degrees Celsius 310 K. Solution. Using the Nernst equation. ED equals RT ZF lane C1 plus C2 plus ED equals ZF RT. Lane C2 plus C1 plus. Given. R equals 8.314. R equals 8.314 J per mole KT. Equals 310 T equals 310 KZ. Equals plus 1. E equals plus 1 for Na F. Equals 96,485. F equals 96,485 C per mole C. 1 plus equals 100 C1 plus. Equals 100 MC. 2 plus equals 150 C2 plus. Equals 150 ME. D equals 8.314 times 310 1 times 96,485 lane 100,150 ED equals 1 times 96,485 8.314 times 310. Lane 150,100 ED equals 2,577.3496. 485 times lane 0.667. ED equals 96,485 2,577.34. Chara and Nutella, green algae, fast cytoplasmic streaming, approximately 50 to 75 microamp, sec, alodia, aquatic plant, chloroplasts move along cell edges, stratoscantia, spiderwort, visible movement of organelles, amoeba, cytoplasmic streaming aids, pseudopodia formation, paramecium helps in intracellular digestion. Solved previous year question. Which cytoskeletal element is primarily responsible for cyclosis? A. Microtubules. B. Actin filaments. C. Intermediate filaments. D. Collagen fibers. Solution. Cyclosis is driven by actin filaments, which serve as tracks for myosin motor proteins. This movement requires ATP hydrolysis. Hence, the correct answer is B. Actin filaments. Multiple choice questions. MCQs. Cyclosis is most commonly observed in which type of cells? A. Bacterial cells. B. Small animal cells. C. Large plant cells. D. Viruses. Answer. C. Large plant cells. Which of the following is not a function of cyclosis? Intracellular transport. B. Photosynthesis optimization. C. Active transport across membranes. D. Wound healing. Answer. C. Active transport across membranes. Active transport is mediated by membrane proteins, not cyclosis. In plant cells, the movement of chloroplasts due to cyclosis primarily helps in plant cells, the movement of chloroplasts due to cyclosis primarily helps in mitosis. B. ATP synthesis. C. Light absorption. D. Protein synthesis. Answer. C. Light absorption. This completes the detailed study material on cyclosis for your PhD entrance exam in zoology. You said, plasmolysis. ChatGPT said, plasmolysis introduction. Plasmolysis is the process of cell shrinkage due to the loss of water from a cytoplasm when a plant cell is placed in a hypertonic solution. This phenomenon is unique to plant cells because of the presence of a cell wall, which remains intact while the protoplasm contracts away from it. Plasmolysis is reversible or irreversible, depending on the intensity and duration of water loss. It is an important concept in plant physiology, helping in understanding osmotic balance, water potential, and membrane permeability. Types of plasmolysis. Incipient plasmolysis. The first stage of plasmolysis where the plasma membrane just starts detaching from a cell wall. Water loss is minimal, and the cell can recover if placed in an isotonic solution. Evident concave plasmolysis. Significant water loss causes complete detachment of the protoplast from the cell wall. The cell remains alive and can regain its original state in a hypotonic solution, a process called deplasmolysis. Irreversible convex plasmolysis. Extreme water loss leads to the destruction of the cytoskeleton and plasma membrane damage. The cell cannot recover and eventually dies. Mechanism of plasmolysis. Hypertonic environment. When a plant cell is placed in a solution with higher solute concentration, e.g., NaCl or sugar solution, water moves out of the cell by osmosis. Loss of turgor pressure. As water exits, the vacuole shrinks, reducing turgor pressure, pressure exerted by the cell membrane against the cell wall. Protoplasm retraction. The plasma membrane detaches from the cell wall, and the cytoplasm shrinks. Formation of plasmolyzed cell. The cell wall remains rigid, while the protoplast contracts into a smaller mass. Factors affecting plasmolysis. Concentration of external solution. A higher solute concentration speeds up plasmolysis. Temperature. High temperatures increase the rate of osmosis, accelerating plasmolysis. Cell type and age. Younger cells with higher water content plasmolyze faster than older cells. Cell wall composition. Thickness and flexibility of the cell wall influence the extent of plasmolysis. Significance of plasmolysis. Determination of osmotic potential. Used to study water potential and solute concentration in plant tissues. Preservation of food. High salt or sugar concentrations cause plasmolysis in bacteria and fungi, preventing food spoilage. Survival mechanism in plants. Some plants undergo plasmolysis as a temporary adaptation to drought, reducing water loss. Agricultural implications. Overuse of chemical fertilizers or irrigation with saline water induces plasmolysis, damaging crops. Examples of plasmolysis in nature. Organism. Example of plasmolysis: onion epidermal cells placed in hypertonic sugar, salt solution. Aquatic plants. Alodia, Spirogyra, exposed to saline water. Bacteria and fungi undergo plasmolysis in highly concentrated environments. Pickling, jams, honey. Solved previous year question. Question. Which of the following best describes plasmolysis? A. Entry of water into a plant cell, making it turgid. B. Shrinkage of cytoplasm due to water loss in a hypertonic solution. C. Bursting of an animal cell when placed in a hypotonic solution. D. Active transport of water molecules across the cell membrane. Solution. Plasmolysis occurs when water moves out of a plant cell due to osmosis, leading to the shrinkage of the cytoplasm. The plasma membrane detaches from a cell wall, but the wall remains intact. Correct answer. B. Shrinkage of cytoplasm due to water loss in a hypertonic solution. Multiple choice questions. MCQs. Which condition is required for plasmolysis to occur? A. Isotonic solution. B. Hypotonic solution. C. Hypertonic solution. D. Distilled water. Answer. C. Hypertonic solution. What happens to the plasma membrane during plasmolysis? A. It remains attached to the cell wall. B. It breaks apart completely. C. It detaches from a cell wall and contracts. D. It becomes thicker. Answer. C. It detaches from a cell wall and contracts. Which of the following processes reverses plasmolysis? A. Exocytosis. B. Diffusion. C. Deplasmolysis. D. Active transport. Answer. C. Deplasmolysis. This concludes the detailed study material on plasmolysis for your PhD entrance exam in zoology. You said, Van Hoff's laws. ChatGPT said, Van Hoff's laws introduction. Van Hoff's laws describe the quantitative relationship between osmotic pressure, temperature, and solute concentration in a solution. These laws are fundamental in physical chemistry and biology, particularly in explaining osmotic behavior in cells. Van Hoff's work laid the foundation for understanding how solute concentration affects osmotic pressure, which is crucial in cell physiology, plant-water balance, and biochemical reactions. Van Hoff's first law, osmotic pressure law. This law states that the osmotic pressure, pi, of a dilute solution is directly proportional to its molar concentration, C, and absolute temperature, T. Mathematically, it is expressed as pi equals CRT, pi equals CRT, where pi equals osmotic pressure in atm or pascals, C equals molar concentration of the solute, mole per liter, R equals universal gas constant, 0.0821 LATM mol KT. T equals absolute temperature in Kelvin. K. Biological significance explains how cells maintain osmotic balance. Helps understand water uptake in plant roots through osmosis. Predicts osmotic pressure in blood plasma and intracellular fluids. Van Hoff's second law, relation to colligative properties. This law states that osmotic pressure is a colligative property, meaning it depends on the number of solute particles rather than their identity. For an electrolyte solution, where solutes dissociate into ions, the osmotic pressure equation is modified using the Van Hoff factor I. Pi equals ICRT. Pi equals ICRT, where I counts for the number of ions produced per molecule of solute. For example, NaCl dissociates into NaCl, so I equals 2. CaCl dissociates into Ca squared and 2Cl, so I Equals 3. I equals 3. Biological significance. Important in plasmolysis and osmoregulation in cells. Explains why electrolyte solutions exert more osmotic pressure than non-electrolyte solutions. Used in medical applications like IV fluid administration and dialysis. Applications of Van Hoff's laws in biology cellular osmoregulation. Explains water movement across membranes in response to solute concentration. Predicts turbo pressure in plant cells, critical for maintaining rigidity. Plant water transport. Van Hoff's laws. ChatGPT said, Van Hoff's laws introduction. Van Hoff's laws describe the quantitative relationship between osmotic pressure, temperature, and solute concentration in a solution. These laws are fundamental in physical chemistry and biology, particularly in explaining osmotic behavior in cells. Van Hoff's work laid the foundation for understanding how solute concentration affects osmotic pressure, which is crucial in cell physiology, plant-water balance, and biochemical reactions. Van Hoff's first law, osmotic pressure law. This law states that the osmotic pressure pi of a dilute solution is directly proportional to its molar concentration C and absolute temperature T. Mathematically, it is expressed as pi equals CRT where pi equals osmotic pressure in atm or pascals C. C equals molar concentration of a solute mole per liter R. R equals universal gas constant 0.0821 LATM mol KT. T equals absolute temperature in Kelvin K. Biological significance explains how cells maintain osmotic balance. Helps understand water uptake in plant roots through osmosis. Predicts osmotic pressure in blood plasma and intracellular fluids. Van Hoff's second law, relation to colligative properties. This law states that osmotic pressure is a colligative property, meaning it depends on the number of solute particles rather than their identity. For an electrolyte solution, where solutes dissociate into ions, the osmotic pressure equation is modified using the Van Hoff factor I. Pi equals ICRT. I equals ICRT where I counts for the number of ions produced per molecule of solute. For example, NaCl dissociates into non-Cl, so I equals 2. I equals 2. CaCl dissociates into Ca squared and 2Cl, so I equals 3. I equals 3. Biological significance. Important in plasmolysis and osmoregulation in cells. Explains why electrolyte solutions exert more osmotic pressure than non-electrolyte solutions. Used in medical applications like IV fluid administration and dialysis. Applications of Van Hoff's laws in biology cellular osmoregulation. Explains water movement across membranes in response to solute concentration. Predicts turbo pressure in plant cells, critical for maintaining rigidity. Plant water transport. Helps in understanding root pressure and water absorption. Determines osmotic potential of xylem sap. Blood plasma in medical applications. Used in designing isotonic solutions for intravenous fluids. Helps in dialysis and blood filtration processes. Biochemical reactions. Important in enzyme activity studies where solute concentration affects reaction rates. Solved previous year question. Question. According to Van Hoff's equation, osmotic pressure is directly proportional to a volume of solution. Heat temperature and molar concentration of solute C, molecular weight of solute D, density of a solution. Solution. Van Hoff's equation states pi equals ICRT. Pi equals ICRT. Where osmotic pressure pi depends on solute concentration C and absolute temperature T. Therefore, the correct answer is B. Temperature and molar concentration of solute. Multiple choice questions MCQs. Van Hoff's law applies to a gaseous mixtures B. Solid state diffusion C. Dilute solutions D. All of the above. Answer C. Dilute solutions. Which factor is used in Van Hoff's equation to account for ion dissociation? A. Avogadro's number. B. Van Hoff factor I. C. Henry's constant D. pH scale. Answer B. Van Hoff factor I. What is the osmotic pressure of a 0.1 MnACl solution at 300 K? Assume complete dissociation. R equals 0.0821 LATM mol K at 2.46 atmospheres. B. 1.23 atmospheres C. 4.92 atmospheres D. 3.69 atmospheres. Solution. Given. C equals 0.1 C equals 0.1 Mi. Equals 2. I equals 2 for NaCl R. Equals 0.0821. R equals 0.0821 LATM mol KT. Equals 300 T equals 300 K. Using Van Hoff's equation. Pi equals ICRT equals 2, 0.1. 0.0821. 300. Pi equals ICRT equals 2, 0.1, 0.0821, 300 pi. Equals 4.92 atmospheres. Pi equals 4.92 atmospheres. Answer, C. 4.92 atmospheres. This concludes the detailed study material on Van Hoff's laws for your PhD entrance exam in zoology. You said. Osmotic concentration. ChatGPT said. Osmotic concentration introduction. Osmotic concentration, also known as osmolarity, is a measure of the total solute concentration in a solution. It is defined as the number of osmoles of solute per liter of solution and is expressed in osmoles per liter (OSML) or milliosmoles per liter (mosm)L). This concept is essential in cell biology, physiology, and biochemistry, as it governs water movement across cell membranes, impacting cell function, osmoregulation, and homeostasis. Types of osmotic concentration. Isoosmotic solution. A solution with the same osmotic concentration as the cell's cytoplasm. No net water movement occurs across the membrane. Hyperosmotic solution. A solution with a higher osmotic concentration than the cell's cytoplasm. Water moves out of the cell, leading to cell shrinkage, plasmolysis in plants or crenation in animal cells. Hypoosmotic solution. A solution with a lower osmotic concentration than the cell's cytoplasm. Water enters the cell, causing it to swell or burst, cytolysis in animal cells or turgidity in plant cells. Measurement of osmotic concentration. 1. Osmolarity versus osmolality. Osmolarity, osmoles of solute per liter of solution, OSML. Osmolality, osmoles of solute per kilogram of solvent, OSM per kilogram. Osmolality is preferred in physiology because it is independent of temperature and pressure. 2. Calculation of osmolarity. For a solution containing a non-electrolyte solute, e.g., glucose, osmolarity equals C times N. Osmolarity equals C times N where C equals molar concentration, mole per liter, N equals number of particles the solute dissociates into. Van Hoff factor for electrolytes, e.g., NaCl, CaCl, dissociation must be considered. Osmolarity equals C times I. Osmolarity equals C times I where I is the Van Hoff factor, number of ions per molecule. Example: NaCl table salt dissociates into Non-Cl, right pointing arrow I equals 2. I equals 2. CaCl calcium chloride dissociates into Ca squared and 2Cl, right pointing arrow I equals 3. I equals 3. Biological importance of osmotic concentration. Osmoregulation in cells. Cells maintain a stable osmotic environment to prevent dehydration or swelling. Important in kidney function, blood plasma regulation, and plant water balance. Turgor pressure in plants. Determines water uptake by plant cells. Regulates stomatal movement and overall plant rigidity. Medical applications. Intravenous IV fluids must be isoosmotic to prevent cell damage. Hyperosmotic or hyposmotic solutions are used in medical treatments, e.g., dehydration therapy. Physiological homeostasis. Blood plasma osmolarity is maintained at approximately 300 mosm for proper cellular function. Disruptions can lead to conditions like dehydration, edema, and electrolyte imbalances. Solved previous year question. Question: which of the following solutions has the highest osmotic concentration? 0.1 m glucose B, 0.1 m NaClC, 0.1 m CaClD, 0.1 m sucrose. Solution: glucose and sucrose do not dissociate. Van Hoff factor I equals 1. I equals 1. NaCl dissociates into Non-Cl I equals 2. I equals 2. CaCl dissociates into Ca squared and 2Cl I equals 3. I equals 3. Using osmolarity equals C times I. Osmolarity equals C times I. Glucose, 0.1. Times 1 equals 0.1. 0.1 times 1 equals 0.1 OSM LNaCl. 0.1 times 2 equals 0.2. 0.1 times 2 equals 0.2 OSM LCaCl. 0.1 times 3 equals 0.3. 0.1 times 3 equals 0.3 OSM L sucrose, 0.1. Times 1 equals 0.1. 0.1 times 1 equals 0.1 OSM L. Correct answer, C. 0.1 MCACl. Multiple choice questions, MCQs. Which of the following describes a hyperosmotic solution? A solution with higher osmotic concentration than the cell. B, a solution with lower osmotic concentration than the cell. C, a solution with the same osmotic concentration as the cell. D, a solution that does not affect water movement. Answer: A solution with higher osmotic concentration than the cell. What is the osmolarity of a 0.2 m NaCl solution? Assume complete dissociation. I equals 2. I equals 2. At 0.1 OSM LB, 0.2 OSM LC, 0.4 OSM LD, 0.6 OSM L. Solution: osmolarity equals C times I equals 0.2 times 2 equals 0.4 OSM L. Osmolarity equals C times I equals 0.2 times 2 equals 0.4 OSM L. Answer: C. 0.4 OSM L. Which of the following body fluids has an osmolarity of approximately 300 mosm L? A. Urine. B. Blood plasma. C. Gastric juice. D. Saliva. Answer: B. Blood plasma. This concludes the detailed study material on osmotic concentration for your PhD entrance exam in zoology. You said, osmotic gradient. ChatGPT said, osmotic gradient introduction. An osmotic gradient refers to the difference in osmotic concentration, osmolarity, between two solutions separated by a semi-permeable membrane. This difference drives the movement of water molecules through osmosis, a fundamental process in cell biology, physiology, and ecology. Cells and organisms use osmotic gradients to regulate water balance, control solute transport, and maintain homeostasis. Understanding osmotic gradients is crucial for biomedical applications, kidney function, plant physiology, and marine biology. Mechanism of osmotic gradient. Water moves from a lower osmotic concentration, hypoosmotic region, to a higher osmotic concentration, hyperosmotic region, until equilibrium is reached. The driving force behind osmosis is the osmotic pressure difference between the two solutions. In biological systems, cell membranes, capillary walls, and organ surfaces act as semi-permeable barriers controlling the movement of water and solutes. Types of osmotic gradients in biological systems. 1. Intracellular versus extracellular fluid gradients. Cells maintain an osmotic gradient between the cytoplasm and extracellular environment. A disturbance in this gradient leads to cell shrinkage, granation, in hypertonic solutions, or cell swelling, lysis, in hypotonic solutions. 2. Kidney and urine concentration. The loop of Henle and nephrons creates a strong osmotic gradient for water reabsorption. The renal medulla maintains a hyperosmotic environment, pulling water out of the collecting ducts to concentrate urine. 3. Plant water transport, root to chute gradient. Roots absorb water from low solute soil water, hypotonic, and transport it upwards to regions with higher solute concentration. The xylem utilizes osmotic gradients and capillary action to move water against gravity. 4. Osmotic gradients in marine and freshwater organisms. Marine fish live in a hyperosmotic environment and constantly lose water, requiring active salt uptake and minimal urination. Freshwater fish live in a hypoosmotic environment and absorb excess water, requiring active salt retention and frequent urination. Mathematical representation of osmotic gradient. The osmotic gradient, delta pi dP, is given by delta pi equals pi inside minus pi outside. dP equals pi inside minus pi outside, where pi inside equals osmotic pressure inside the cell or system, pi outside equals osmotic pressure outside the cell or system A higher. Delta. Pi. dP means greater water movement across the membrane. Biological importance of osmotic gradient: maintains cell volume and function. Prevents cell shrinkage or bursting due to uncontrolled water movement. Regulates ion exchange and nutrient uptake. Regulates blood pressure and fluid balance. Plasma osmolarity determines fluid exchange between blood vessels and tissues. Disturbances can lead to edema, dehydration, or circulatory shock. Essential for kidney filtration. The glomerulus creates an osmotic gradient to filter blood plasma. The collecting ducts use gradients to retain or excrete water, regulating urine concentration. Drives nutrient absorption in intestines. The small intestine absorbs nutrients by creating local osmotic gradients, ensuring efficient water and solute transport. Enables marine and freshwater adaptations. Osmoregulation in aquatic organisms relies on maintaining osmotic gradients between internal fluids and external water. Solved previous year question. Question. Which of the following structures in the kidney is responsible for maintaining an osmotic gradient essential for water reabsorption? A. Glomerulus. B. Loop of Henle. C. Bowman's capsule. D. Proximal convoluted tubule. Solution. The loop of Henle creates an osmotic gradient in the renal medulla, allowing water reabsorption from the collecting ducts. This mechanism is vital for urine concentration and fluid homeostasis. Correct answer, B. Loop of Henle. Multiple choice questions, MCQs. Water moves across a semi-permeable membrane due to an osmotic gradient. Which direction does water move? Up from hyperesmotic to hypoosmotic side. B. From hypoosmotic to hyperesmotic side. C. Randomly in both directions. D. Against the osmotic gradient. Answer, B. From hypoosmotic to hyperesmotic side. Which organ system in humans heavily depends on osmotic gradients for its function? A. Digestive system. B. Endocrine system. C. Urinary system. D. Muscular system. Answer, C. Urinary system. Which marine animals drink seawater to compensate for osmotic water loss? A. Freshwater fish. B. Amphibians. C. Marine bony fish. D. Mammals. Answer, C. Marine bony fish. This concludes the detailed study material on osmotic gradient for your PhD entrance exam in zoology. You said, electroosmosis. ChatGPT said, electroosmosis introduction. Electroosmosis is the movement of a liquid through a porous medium or a capillary due to an applied electric field. This phenomenon occurs because charged surfaces attract counterions, forming an electric double layer at the interface, which interacts with the applied field to drive fluid movement. Electroosmosis is widely applied in biological systems, analytical chemistry, biomedical devices, and soil engineering. It plays a crucial role in membrane transport, microfluidics, and electrophoretic separation techniques. Mechanism of electroosmosis: formation of the electric double layer, EDL. When an electrolyte comes in contact with a charged solid surface, e.g., cell membranes, capillaries, or gel pores, ions from the solution arrange themselves near the surface. This creates a diffuse electric double layer, EDL, with opposite charges in solution balancing the surface charge. Application of an electric field. When an external electric potential is applied across a system, ions in the diffuse layer move, dragging solvent molecules along due to viscous forces. This results in bulk liquid movement known as electroosmotic flow, EOF. Electroosmotic flow profile. Unlike pressure-driven flow, Poiseuille flow, which is parabolic, EOF has a nearly uniform, plug-like velocity profile due to the dragging of fluid by the charged layer. This makes EOF highly useful in microfluidic and capillary electrophoresis applications, as it minimizes dispersion. Mathematical representation of electroosmosis. The electroosmotic velocity P, EO is given by versus EO equals epsilon zeta E eta V EO equals zeta as E, where epsilon equals permittivity of the medium zeta. Zeta equals zeta potential, electric potential at the sliving plane of the EDL. E equals applied electric field, B per meter eta. Eta equals viscosity of the fluid. A higher zeta potential or electric field increases the electroosmotic flow. Biological and practical applications of electroosmosis: capillary electrophoresis, CE. Electroosmotic flow is exploited in capillary electrophoresis to transport samples through narrow capillaries. It allows efficient separation of biomolecules, including proteins, nucleic acids, and drugs. Membrane transport in cells. In ion channels and porous membranes, electroosmosis assists in solute transport and cell signaling. It contributes to nerve impulse transmission and bioelectrical phenomena. Microfluidic lab on A-chip devices. Electroosmotic flow enables precise fluid control in microchannels, used in DNA sequencing, PCR, and biosensors. It eliminates the need for mechanical pumps, making devices compact and efficient. Soil decontamination and water removal. Electroosmosis is applied in geotechnical engineering to dewater fine-grained soils and remove contaminants. Used in groundwater purification and industrial wastewater treatment. Drug delivery systems. Electroosmotic principles are used in iontophoresis, a technique that delivers drugs through the skin using an electric field. Enhances the penetration of charged pharmaceutical molecules into tissues. Bioelectrochemical systems. Electroosmosis plays a role in biofuel cells and biosensors, where ion migration under electric fields influences biochemical reactions. Solved previous year question. Question. Which factor primarily affects the rate of electroosmotic flow? A. Surface charge of the solid phase. B. Viscosity of the liquid. C. Strength of the applied electric field. D. All of the above. Solution. The surface charge influences the formation of the electric double layer. Viscosity affects how easily fluid moves under an electric field. Stronger electric fields enhance electroosmotic velocity. Since all factors contribute to the electroosmotic flow, the correct answer is D. All of the above. Multiple choice questions, MCQs. Which of the following best describes electroosmosis? A. Movement of charged particles in an electric field. B. Movement of fluid through a charged surface under an electric field. C. Movement of water due to osmosis. D. Movement of solvent due to concentration gradients. Answer, B. Movement of fluid through a charged surface under an electric field. Which of the following applications relies on electroosmotic flow? A. Capillary electrophoresis. B. Kidney filtration. C. Passive diffusion across membranes. D. Active transport in cells. Answer, A. Capillary electrophoresis. Which parameter does not directly influence electroosmotic flow? A. Zeta potential. B. Applied electric field. C. Molecular weight of solute. D. Viscosity of the liquid. Answer: C. Molecular weight of solute. This concludes the detailed study material on electroosmosis for your PhD entrance exam in zoology. You said. Electrolytic and ionic balance in biological fluid. ChatGPT said. Electrolytic and ionic balance in biological fluids. Introduction. Electrolytic and ionic balance in biological fluids is essential for cellular homeostasis, nerve transmission, muscle function, and enzymatic activity. Biological fluids, including blood, intracellular fluid (ICF) and extracellular fluid (ECF), maintain specific concentrations of ions, electrolytes such as sodium N, A+, Na+, potassium K+, K+, calcium C, A2+, Ca2+, chloride C, L-Cl-, bicarbonate H, CO3-H, CO3-, and phosphate P, O4-3-, PO4-3-. The maintenance of ionic balance is regulated by osmosis, diffusion, active transport, and buffering systems. Disruptions in ionic homeostasis can lead to neurological disorders, dehydration, kidney dysfunction, and acid-base imbalances. Major electrolytes in biological fluids and their functions: electrolyte major location, primary function sodium N, A+, Na+, extracellular fluid ECF, nerve impulse transmission, osmotic balance, blood pressure regulation, potassium K+, K+, intracellular fluid ICF, membrane potential, muscle contraction, enzyme activation, calcium C, A2+, Ca2+, ECF and ICF, bone formation, neurotransmitter release, muscle contraction, blood clotting, chloride C, L-Cl-, ECF maintains osmotic balance, acid-base balance, gastric acid formation, bicarbonate H, CO3-H, CO3-. ECF maintains pH balance, carbon dioxide transport, phosphate P, O4-3-, PO4-3-. ICF ATP production, nucleic acid synthesis, buffering, magnesium M, G2+, Mg2+. ICF cofactor for enzymes, muscle and nerve function mechanisms regulating ionic balance. 1. Role of cell membranes in ionic homeostasis. Plasma membranes are selectively permeable and regulate ion movement through channels, pumps, and transporters. Sodium-potassium pump Na KATPase actively pumps 3 Na out and 2 K in, maintaining the resting membrane potential. Calcium pumps and exchangers regulate intracellular C, A2+, Ca2+. Levels, preventing cytotoxicity. 2. Osmotic balance and water distribution. Osmotic pressure is maintained by ion concentration gradients between intracellular and extracellular fluids. Hypertonic conditions cause water loss from cells shrinking, while hypotonic conditions cause water influx, swelling. Albumin and electrolytes regulate oncotic pressure, keeping fluid balance in blood vessels. 3. Role of the kidney in electrolyte regulation. The nephrons filter electrolytes and regulate their reabsorption or excretion. Sodium and water balance, controlled by aldosterone, increases NA reabsorption, and antidiuretic hormone ADH promotes water retention. Potassium balance, excess K+, K+, is excreted through the urine, preventing hyperkalemia. Acid-base balance, bicarbonate H, CO3-H, CO3-, is regulated to maintain blood pH 7.35-7.45. 4. Acid-base balance, buffer systems. Bicarbonate buffer system maintains blood pH by balancing H, CO3-H, CO3-, and H+. Phosphate buffer system regulates pH inside cells and in urine. Protein buffer system, hemoglobin and plasma proteins act as pH buffers in the blood. Disorders related to electrolyte imbalance. Disorder, electrolyte imbalance symptoms: hyponatremia, low N, A+, Na+. Confusion, seizures, coma, hypernatremia, high N, A+, Na+. Thirst, agitation, dehydration, hypokalemia, low K+, K+. Muscle weakness, arrhythmias, hyperkalemia, high K+, K+. Cardiac arrest, paralysis, hypocalcemia, low C, A2+, Ca2+. Tetany, muscle cramps, osteoporosis, hypercalcemia, high C, A2+, Ca2+. Kidney stones, confusion, nausea. Solved previous year question. Which hormone regulates sodium and potassium balance in the kidneys? A. Insulin. B. Aldosterone. C. Adrenaline. D. Glucagon. Solution. Aldosterone, secreted by the adrenal cortex, promotes sodium reabsorption and potassium excretion in the kidneys, maintaining electrolyte balance. Correct answer, B. Aldosterone. Multiple choice questions, MCQs. Which ion is the primary determinant of extracellular osmolarity? A. Potassium. K+, K+. B. Sodium. N. A+, Na+. C. Calcium. C. A2+, Ca2+. D. Magnesium. M. G2+, Mg2+. Answer, B. Sodium. N. A+, Na+. Which organ is primarily responsible for maintaining electrolyte balance in the body? A. Liver. B. Kidney. C. Pancreas. D. Heart. Answer, B. Kidney. Which buffer system plays a major role in maintaining blood pH? A. Phosphate buffer. B. Protein buffer. C. Bicarbonate buffer. D. Sulfate buffer. Answer, C. Bicarbonate buffer. This concludes the detailed study material on electrolytic and ionic balance in biological fluids for your PhD entrance exam in zoology. You said. Viscosity and factors affecting viscosity. ChatGPT said. Viscosity and factors affecting viscosity. Introduction. Viscosity is the measure of a fluid's resistance to flow. It determines how easily a fluid can deform under an applied force. In biological and physiological systems, viscosity plays a crucial role in blood circulation, cytoplasmic streaming, lubrication in joints, and intracellular transport. Viscosity results from intermolecular forces and varies with temperature, pressure, and fluid composition. It is categorized as dynamic viscosity, eta. Eta, the internal friction within a fluid, measured in poise, P, or Pascal seconds, Pa s. Kinematic viscosity, nu. Note the ratio of dynamic viscosity to density, measured in Stokes' Street. Factors affecting viscosity. 1. Temperature. In liquids, viscosity decreases with increasing temperature because molecular interactions weaken, allowing easier movement. In gases, viscosity increases with temperature due to increased molecular collisions. 2. Pressure. In liquids, viscosity increases with pressure because molecular density increases, restricting flow. In gases, viscosity is independent of moderate pressure changes, as gas molecules remain widely spaced. 3. Molecular size and shape. Larger molecules or long-chain polymers have higher viscosity due to stronger intermolecular forces and entanglement. Spherical molecules have lower viscosity compared to elongated molecules. 4. Intermolecular forces. Fluids with strong hydrogen bonding, e.g., water, glycerol, have higher viscosity than nonpolar fluids. Ionic interactions can also increase viscosity, as seen in electrolyte solutions. 5. Solute concentration. Higher solute concentration increases viscosity, as dissolved particles create resistance to flow, e.g., protein-rich plasma in blood. 6. Shear rate. Non-Newtonian fluids. Newtonian fluids, e.g., water, air, have constant viscosity regardless of shear force. Non-Newtonian fluids, e.g., blood, cytoplasm, change viscosity with applied force. Types include shear thinning, pseudoplastic, viscosity decreases with shear stress, e.g., blood, paint. Shear thickening, dilatant, viscosity increases with shear stress, e.g., cornstarch in water. Bingham plastics require a minimum force to flow, e.g., toothpaste, biological gels. 7. Presence of suspended particles. Fluids containing suspended cells, proteins, or colloidal particles, e.g., blood, synovial fluid, have higher viscosity. Viscosity depends on particle size, shape, and concentration. 8. Biological and physiological conditions. Blood viscosity is influenced by hematocrit level, RBC concentration, plasma proteins, and temperature. Synovial fluid viscosity is regulated by hyaluronic acid, providing joint lubrication. Cytoplasmic viscosity affects intracellular transport and organelle movement. Applications of viscosity in biology. Blood flow and circulation. Blood viscosity increases cardiovascular stress, e.g., in dehydration, polycythemia. Low viscosity can cause excessive bleeding, e.g., anemia, hemophilia. Cytoplasmic streaming. Viscosity influences organelle transport and nutrient distribution in cells. Synovial fluid and joint lubrication. Viscosity changes in arthritis lead to joint pain and stiffness. Biotechnological and industrial applications. Used in designing drug formulations, artificial blood, and biopolymer solutions. Solved previous year question. Question. Which of the following factors decreases the viscosity of blood? A. Increased red blood cell concentration. B. High plasma protein level. C. Dehydration. D. Increased temperature. Solution. Higher temperature reduces intermolecular interactions, lowering blood viscosity. Correct answer, D. Increased temperature. Multiple choice questions, MCQs. How does viscosity change with temperature in liquids? A. Increases. B. Decreases. C. Remains constant. D. First increases, then decreases. Answer, B. Decreases. Which of the following is an example of a shear thinning fluid? A. Water. B. Blood. C. Cornstarch in water. D. Mercury. Answer, B. Blood. Which biological fluid's viscosity is influenced by hyaluronic acid? A. Blood. B. Synovial fluid. C. Cerebrospinal fluid. D. Urine. Answer, B. Synovial fluid. This concludes the detailed study material on viscosity and its factors for your PhD entrance exam in zoology. This concludes the detailed study material on viscosity and its factors for your PhD entrance exam in zoology. You said. Determination of viscosity of liquids. ChatGPT said. Determination of viscosity of liquids. Introduction. Viscosity is the measure of a liquid's resistance to flow. It determines the internal friction between adjacent layers of the fluid and is a crucial property in biological, industrial, and chemical applications. The measurement of viscosity is essential in blood flow studies, pharmaceutical formulations, and biochemical reactions. Types of viscosity. Dynamic viscosity. Eta. Eta, the force required to move one layer of fluid over another. Measured in poise P or Pascal seconds Pa s. Kinematic viscosity. Nu. Nu, the ratio of dynamic viscosity to density, measured in Stokes' Street or square meter per second. Methods for viscosity determination. 1. Capillary flow method. Ostwald's viscometer principle. Based on Poiseuille's law, which states that the volume of liquid flowing through a capillary tube is proportional to its viscosity. The time taken for a fixed volume of liquid to pass through a capillary under gravity is measured. Formula. Eta 1 equals eta 2 times rho 1 T1 rho 2 T2. Eta 1 equals eta 2 times rho 2 T2. Rho 1 T1. Where eta 1, eta 2, eta 1, eta 2 equals viscosities of unknown and reference liquids rho 1, rho 2, rho 1, rho 2 equals densities of unknown and reference liquids T 1, T2, T1, T2 equals flow times of unknown and reference liquids. Applications. Used for low viscosity fluids like blood, plasma, and water-based solutions. 2. Falling sphere method. Stokes' law method principle. A small sphere falls through a liquid under gravity. The velocity of the falling sphere is used to calculate viscosity based on Stokes' law. Formula. Eta equals 2 9 R2 rho s minus rho f G. P eta equals 9 2 V R2 rho s minus rho f G. Where R equals radius of the sphere rho s rho f equals densities of sphere and fluid G. G equals gravitational acceleration V. V equals terminal velocity of the sphere. Applications. Used for biological fluids like synovial fluid and cytoplasmic viscosity measurement. 3. Rotational viscometer. Brookfield viscometer principle. A rotating spindle is immersed in a liquid, and the torque required to rotate it is proportional to viscosity. Applications. Used for non-Newtonian fluids like blood, polymer solutions, and gels. 4. Cone and plate viscometer. Principle. A cone is rotated against a flat plate, and the resistance to motion is measured. Applications. Used for low-volume samples in biochemical and pharmaceutical applications. Solved previous year question. Question. Which instrument is used to measure the viscosity of Newtonian fluids like water? A. Rotational viscometer. B. Ostwald's viscometer. C. Falling sphere viscometer. D. Cone and plate viscometer. Solution. Ostwald's viscometer is used for Newtonian fluids like water. Correct answer, B. Ostwald's viscometer. Multiple choice questions, MCQs. Which law is used in the falling sphere method of viscosity measurement? A. Poiseuille's law. B. Stokes' law. C. Newton's second law. D. Boyle's law. Answer, B. Stokes' law. Which viscometer is commonly used for measuring the viscosity of polymer solutions? A. Brookfield viscometer. B. Ostwald's viscometer. C. U-tube viscometer. D. Falling sphere viscometer. Answer, A. Brookfield viscometer. What happens to viscosity when temperature increases in a liquid? A. Increases. B. Decreases. C. Remains constant. D. First increases, then decreases. Answer, B. Decreases. This concludes the detailed study material on the determination of viscosity of liquids for your PhD entrance exam in zoology. You said, significance. ChatGPT said. Significance of viscosity in biological systems. Introduction. Viscosity plays a fundamental role in biological systems, influencing processes at cellular, physiological, and ecological levels. It affects blood flow, nutrient transport, intracellular movement, and biomolecular interactions. Understanding viscosity is essential for fields like medicine, biotechnology, and environmental biology. 1. Importance in blood circulation and cardiovascular health. Blood viscosity affects blood pressure, circulation efficiency, and oxygen delivery. Higher viscosity due to dehydration, polysemia increases cardiac workload, leading to conditions like hypertension and stroke. Lower viscosity, e.g., anemia, hemophilia, results in poor oxygen transport and excessive bleeding. Used in diagnosing blood disorders like sickle cell anemia and hyperviscosity syndromes. 2. Role in cytoplasmic and intracellular transport. Cytoplasm has a viscoelastic nature, affecting organelle movement and biochemical reactions. High cytoplasmic viscosity slows down diffusion and intracellular transport, affecting processes like protein folding and enzyme activity. Cytoplasmic streaming cyclosis depends on optimal viscosity for efficient nutrient and waste transport. 3. Synovial fluid and joint lubrication. Synovial fluid viscosity is crucial for joint movement and shock absorption. Hyaluronic acid maintains viscosity, and a reduction leads to arthritis and joint pain. Viscosity-modifying drugs, e.g., hyaluronic acid injections, are used in osteoarthritis treatment. 4. Importance in respiratory function. Mucosal secretions in the respiratory tract have optimal viscosity for trapping pathogens and facilitating ciliary movement. Increased mucus viscosity, e.g., cystic fibrosis, leads to breathing difficulties and infection risk. Mucolytic drugs target viscosity to improve respiratory health. 5. Digestive system and gastrointestinal health. Viscosity affects gastric emptying, enzyme diffusion, and nutrient absorption. High viscosity in food, e.g., fiber-rich diets, slows digestion, promoting better glucose regulation and satiety. Low viscosity fluids in the gut facilitate rapid digestion and absorption. 6. Importance in drug formulation and biotechnology. Pharmaceutical drugs, e.g., injectable solutions, eye drops, gels, must have optimal viscosity for effectiveness. Biotechnology applications involve controlled viscosity in bioreactors, tissue engineering, and drug delivery systems. 7. Environmental and ecological significance. Water viscosity influences plankton movement, aquatic organism locomotion, and sedimentation rates. Changes in viscosity due to temperature or pollution affect marine ecosystems. Solved previous year question. Question. Which of the following is a condition where blood viscosity increases abnormally? A. Anemia. B. Polysemia. C. Hemophilia. D. Leukopenia. Solution. In polysemia, an increase in red blood cell concentration leads to higher blood viscosity, affecting circulation. Correct answer, B. Polysemia. Multiple choice questions, MCQs. Which fluid's viscosity is maintained by hyaluronic acid? A. Blood. B. Synovial fluid. C. Cerebrospinal fluid. D. Urine. Answer, B. Synovial fluid. Why is viscosity important in respiratory function? A. It prevents mucus production. B. It helps trap pathogens and facilitates ciliary movement. C. It increases oxygen diffusion. D. It enhances gas exchange in alveoli. Answer, B. It helps trap pathogens and facilitates ciliary movement. What happens when blood viscosity is too high? A. Increased oxygen transport. B. Decreased cardiac workload. C. Increased risk of hypertension and clotting. D. Reduced red blood cell count. Answer, C. Increased risk of hypertension and clotting. This concludes the detailed study material on the significance of viscosity in biology for your PhD entrance exam in zoology. Search. Microscopy is an indispensable tool in zoological research, enabling scientists to observe and analyze structures that are invisible to the naked eye. The evolution of microscopic techniques has significantly advanced our understanding of animal morphology, physiology, and developmental biology. This comprehensive overview delves into various microscopy methods, their principles, applications, and relevance in zoological studies. 1. Optical microscopy. Optical microscopy, also known as light microscopy, utilizes visible light to magnify specimens. It encompasses several techniques. Bright field microscopy, the most conventional form, where light passes directly through the specimen. It's suitable for observing stained or naturally pigmented samples but offers limited contrast for transparent specimens. Dark field microscopy employs oblique illumination, causing light to scatter and making the specimen appear bright against a dark background. This technique enhances contrast in unstained, transparent samples, revealing fine structural details. Phase contrast microscopy, introduced by Fritz Cernick, this method converts phase shifts in light passing through transparent specimens into variations in intensity. It's particularly useful for examining live cells and tissues without staining. Differential interference contrast DIC microscopy, also known as Nomarski microscopy, DIC uses polarized light to produce high-contrast images with a pseudo-3D effect, aiding in the visualization of fine structural details in live specimens. Fluorescence microscopy relies on the emission of light by fluorophores within the specimen when excited by specific wavelengths. This technique is invaluable for localizing specific proteins, nucleic acids, and other molecules, often using fluorescent dyes or genetically encoded markers. 2. Confocal laser scanning microscopy, CLSM. CLSM enhances optical microscopy by using laser light to scan specimens point by point, constructing high-resolution images with improved contrast. It allows for optical sectioning, enabling the reconstruction of three-dimensional structures, which is crucial for studying complex tissues and embryonic development in zoology. 3. Electron microscopy. Electron microscopy offers magnifications up to 2 million times, surpassing the limits of light microscopy. It includes transmission electron microscopy. TEM transmits electrons through ultra-thin specimens, revealing internal structures at the molecular level. TEM is essential for examining cell organelles, viruses, and macromolecular complexes. Scanning electron microscopy (SEM) scans the specimen's surface with a focused electron beam, producing detailed three-dimensional images. SEM is used to study surface morphology and topography of specimens, such as insect exoskeletons and tissue surfaces. 4. Scanning probe microscopy. This category includes techniques like atomic force microscopy (AFM), which measures forces between a sharp probe and the specimen surface to generate topographical maps at the atomic level. AFM is instrumental in studying the mechanical properties of biological specimens, including cell membranes and biomolecules. 5. Sample preparation techniques. Proper specimen preparation is vital for accurate microscopy. Common methods include fixation, preserves tissue morphology and molecular integrity, typically using chemical agents like formaldehyde. Embedding, involves infiltrating tissues with media such as paraffin or resins to provide support during sectioning. The paraffin method is widely used for preparing thin sections for light microscopy. Alternatively, the cell-laden method is employed for larger, harder tissues, while the freezing method is used for rapid sectioning without chemical fixation. N. Wikipedia, org. Sectioning, slicing embedded tissues into thin sections using microdomes or ultramicrodomes, essential for TEM analysis. Staining, applying dyes that bind to specific cellular components, enhancing contrast and enabling differentiation of structures. For instance, hematoxylin and eosin H&E staining is commonly used to distinguish between nuclear and cytoplasmic components. 6. Advanced techniques. Modern microscopy has evolved to include sophisticated methods. Live cell imaging utilizes time-lapse microscopy to observe dynamic processes in living cells, such as cell division, motility, and intracellular transport. Super-resolution microscopy breaks the diffraction limit of light, achieving nanometer-scale resolution. Techniques like stimulated emission depletion (STED) and photoactivated localization microscopy (POM) fall into this category, allowing visualization of molecular interactions at the nanoscale. Correlative light and electron microscopy (CLEM) combines the advantages of light and electron microscopy to correlate dynamic live cell imaging with high-resolution structural information. 7. Applications in zoology. Microscopy techniques are pivotal in various zoological research areas. Taxonomy and systematics. Detailed morphological studies aid in species identification and classification. Developmental biology. Observing embryonic development stages provides insights into growth patterns and congenital anomalies. Histology and cytology. Examining tissues and cells elucidates normal physiology and pathological conditions. Neurobiology. Investigating nervous system structures and functions enhances understanding of neural networks and behavior. Parasitology. Identifying and studying parasites at the microscopic level informs disease control and prevention strategies. 8. Solved previous year question. Question. Describe the principle and applications of fluorescence microscopy in zoological research. Answer. Fluorescence microscopy operates on the principle that certain substances emit light upon excitation by specific wavelengths. In this technique, specimens are labeled with fluorophores, which absorb light at one wavelength excitation. Sources. You said. Visualization of cells and subcellular components by light microscopy. ChatGPT said. Visualizing cells and their subcellular components through light microscopy is fundamental in zoological research, providing insights into cellular architecture and function. This comprehensive overview explores various light microscopy techniques, their principles, and applications in studying cells and subcellular structures. 1. Bright field microscopy. Bright field microscopy is the most conventional form of light microscopy, where light passes directly through the specimen. This technique is suitable for observing stained or naturally pigmented samples, offering clear images of cell morphology and tissue architecture. However, it provides limited contrast for transparent specimens, necessitating staining procedures to enhance visibility. 2. Phase contrast microscopy. Developed by Fritz Cernick, phase contrast microscopy converts phase shifts in light passing through transparent specimens into variations in intensity. This method is particularly advantageous for examining live, unstained cells, allowing researchers to observe dynamic processes such as cell division and motility without altering the specimen. 3. Differential interference contrast (DIC) microscopy. Also known as Nomarski microscopy, DIC employs polarized light to produce high-contrast images with a pseudo-three-dimensional effect. This technique enhances the visualization of fine structural details in live specimens, such as organelles and cytoskeletal elements, by detecting differences in refractive indices within the cell. 4. Fluorescence microscopy. Fluorescence microscopy relies on the emission of light by fluorophores within the specimen when excited by specific wavelengths. This technique is invaluable for localizing specific proteins, nucleic acids, and other molecules within cells. By using fluorescent dyes or genetically encoded markers like green fluorescent protein (GFP), researchers can study the distribution and dynamics of subcellular components with high specificity and sensitivity. 5. Confocal laser scanning microscopy (CLSM). CLSM enhances optical microscopy by using laser light to scan specimens point by point, constructing high-resolution images with improved contrast. It allows for optical sectioning, enabling the reconstruction of three-dimensional structures, which is crucial for studying complex tissues and cellular interactions in zoology. 6. Sample preparation techniques. Proper specimen preparation is vital for accurate visualization under light microscopy. Common methods include fixation, preserves tissue morphology and molecular integrity, typically using chemical agents like formaldehyde. Embedding, infiltrating tissues with media such as paraffin or resins to provide support during sectioning. Sectioning, slicing embedded tissues into thin sections using microdomes, essential for observing internal structures. Staining, applying dyes that bind to specific cellular components, enhancing contrast and enabling differentiation of structures. For instance, hematoxylin and eosin H&E staining is commonly used to distinguish between nuclear and cytoplasmic components. 7. Advanced techniques. Modern light microscopy has evolved to include sophisticated methods. Live cell imaging utilizes time-lapse microscopy to observe dynamic processes in living cells, such as intracellular transport and signal transduction, providing real-time insights into cellular function. Super-resolution microscopy techniques like stimulated emission depletion (STED) and photoactivated localization microscopy (POM) surpass the diffraction limit of light, achieving nanometer scale resolution. These methods allow visualization of molecular interactions at the nanoscale, offering unprecedented detail of subcellular structures. Lattice light sheet microscopy. This technique uses a thin sheet of light to illuminate specimens, reducing phototoxicity and enabling high-speed, high-resolution imaging of live cells in 3 dimensions. It is particularly useful for observing rapid cellular processes with minimal damage to the specimen. 8. Applications in zoology. Light microscopy techniques are pivotal in various zoological research areas. Developmental biology. Observing embryonic development stages provides insights into growth patterns and congenital anomalies. Histology and cytology. Examining tissues and cells elucidates normal physiology and pathological conditions. Biology. Investigating nervous system structures and functions enhances understanding of neural networks and behavior. Parasitology. Identifying and studying parasites at the microscopic level informs disease control and prevention strategies. 9. Solved previous year question. Question. Explain the principle and applications of phase contrast microscopy in visualizing live cells. Answer. Phase contrast microscopy operates on the principle of converting phase shifts in light passing through a transparent specimen into variations in intensity, producing high contrast images without the need for staining. This is achieved by a phase plate that alters the phase of light passing through different parts of the specimen, enhancing contrast. It is particularly useful for observing live cells, allowing researchers to study dynamic processes such as cell division, motility, and intracellular transport in their natural state without chemical fixation or staining. 10. Multiple choice questions. Which microscopy technique utilizes polarized light to produce high contrast images with a pseudo 3-dimensional effect? A. Bright field microscopy. B. Phase contrast microscopy. C. Differential interference contrast DIC microscopy. D. Fluorescence microscopy. Answer. C. Differential interference contrast DIC microscopy. Which of the following is a significant advantage of fluorescence microscopy in cell visualization? A. Ability to observe live cells without staining. B. Enhanced contrast for transparent specimens. C. Specific localization of proteins and nucleic acids within cells. D. Visualization of surface morphology in 3 dimensions. Answer. C. Specific localization of proteins and nuclei. Sources. You said. Resolving powers of different microscopes, ChatGPT said. The resolving power of a microscope is its ability to distinguish between 2 closely spaced points as separate entities, thereby revealing fine structural details. This capability is crucial in zoological research, where understanding cellular and subcellular structures is essential. Different types of microscopes offer varying resolving powers, primarily influenced by the wavelength of the illumination source and the numerical aperture of the objective lens. 1. Optical light microscopes. Optical microscopes utilize visible light to illuminate specimens. The theoretical resolution limit of these microscopes is approximately 0.2 micrometers, 200 nanometers, constrained by the diffraction limit of light. This means that 2 points closer than 200 nanometers cannot be resolved as distinct entities. Advanced techniques, such as confocal laser scanning microscopy, can enhance image contrast and clarity but remain subject to this diffraction limit. MICROSCOPYU, com. 2. Electron microscopes. Electron microscopes employ electron beams with much shorter wavelengths than visible light, enabling significantly higher resolving powers. There are 2 primary types. Transmission electron microscopes TEMs can achieve resolutions up to 0.05 nanometers, 50 picometers, allowing visualization of individual atoms. N. Wikipedia, org. Scanning electron microscopes SEMs provide detailed 3-dimensional images of specimen surfaces with resolutions around 0.4 nanometers, 400 picometers. N. Wikipedia, org. 3. Scanning probe microscopes. Scanning probe microscopes, including atomic force microscopes AFM, can achieve atomic level resolution, enabling visualization of individual atoms and molecular structures. These microscopes are instrumental in nanotechnology and materials science research. 4. Factors influencing resolving power. The resolving power of a microscope is influenced by several factors. Wavelength of illumination. Shorter wavelengths yield higher resolution. For instance, ultraviolet light offers better resolution than visible light in optical microscopy. MICROSCOPYU, com. Numerical aperture NA. A higher NA indicates a greater ability to gather light and resolve fine details. It depends on the refractive index of the medium between the lens and the specimen and the half angle of the maximum cone of light that can enter the lens. Lens quality and aberrations. Imperfections in lens design or manufacturing can reduce resolving power by introducing aberrations that distort the image. 5. Applications in zoology. Understanding the resolving power of various microscopes allows zoologists to select appropriate instruments for their research needs. Light microscopy, suitable for observing live cells, tissues, and organisms, especially when combined with staining techniques to enhance contrast. Electron microscopy, ideal for detailed examination of ultrastructural components, such as organelles, viruses, and macromolecular complexes, providing insights into cellular architecture and function. Scanning probe microscopy, utilized for studying surface topography at the molecular and atomic levels, contributing to understanding of molecular interactions and structural biology. 6. Solved previous year question. Question. Compare and contrast the resolving powers of light microscopes and electron microscopes, and discuss their implications in biological research. Answer. Light microscopes have a resolving power limited to approximately 200 nanometers due to the diffraction limit of visible light, making them suitable for observing cells and larger organelles. In contrast, electron microscopes, utilizing electron beams with shorter wavelengths, achieve much higher resolutions, TEMs up to 0.05 nanometers and SEMs around 0.4 nanometers, allowing visualization of subcellular structures and macromolecules. The higher resolving power of electron microscopes enables detailed structural analysis at the molecular and atomic levels, which is crucial for understanding complex biological processes. 7. Multiple choice questions. Which factor primarily limits the resolving power of a light microscope? A. Numerical aperture of the objective lens. B. Wavelength of the illumination source. C. Quality of the specimen preparation. D. Magnification power of the eyepiece. Answer, beat wavelength of the illumination source. What is the approximate resolving power of a transmission electron microscope TEM? A 200 nanometers. B 0.4 nanometers. C 0.05 nanometers. D 2 micrometers. Answer, C 0.05 nanometers. Which type of microscope is most suitable for observing the surface topology of a specimen at atomic resolution? A. Light microscope. B. Transmission electron microscope. C. Scanning electron microscope. D. Scanning probe microscope. Answer, D. Scanning probe microscope. Understanding the resolving powers of different microscopes is essential for selecting the appropriate imaging technique in zoological research, ensuring that the chosen method aligns with the specific structural details under investigation. Sources. You said. Microscopy of living cells, ChatGPT said. Visualizing living cells through microscopy is pivotal in zoological research, enabling the observation of dynamic biological processes in real time. This comprehensive overview delves into various microscopy techniques tailored for live cell imaging, their principles, applications, and considerations for optimal use. 1. Wide field microscopy. Wide field microscopy is a fundamental technique where the entire specimen is illuminated uniformly, allowing for the observation of live cells with minimal phototoxicity. This method is advantageous for capturing rapid events and is often employed in conjunction with fluorescence imaging to study specific cellular components. However, out-of-focus light can reduce image clarity, making it less suitable for thick specimens. Leica Microsystems, Com. 2. Phase contrast microscopy. Phase contrast microscopy enhances the contrast of transparent specimens without the need for staining, making it ideal for observing live cells. It converts phase shifts in light passing through the specimen into intensity variations, rendering detailed images of cellular structures and processes. This technique is particularly useful for monitoring cell division, motility, and morphological changes. N. Wikipedia, org. 3. Differential interference contrast DIC microscopy. DIC microscopy employs polarized light to produce high-contrast images with a pseudo-three-dimensional appearance. It is beneficial for visualizing fine details in live, unstained cells, such as organelles and cytoskeletal elements. DIC is especially useful in studying cell morphology and intracellular structures with enhanced depth perception. The Scientist, Com. 4. Fluorescence microscopy. Fluorescence microscopy involves labeling specific cellular components with fluorescent probes, allowing for the visualization of targeted structures within live cells. While this technique provides high specificity, it requires careful consideration of phototoxicity and photobleaching, which can affect cell viability and fluorescence intensity over time. Optimizing excitation light intensity and exposure time is crucial to minimize these effects. Leica Microsystems, Com. 5. Confocal laser scanning microscopy, CLSM. CLSM enhances fluorescence microscopy by using laser light to scan the specimen point by point, reducing out-of-focus light and improving image resolution. CLSM enhances fluorescence microscopy by using laser light to scan the specimen point by point, reducing out-of-focus light and improving image resolution. This technique enables optical sectioning, allowing for the reconstruction of three-dimensional structures in live cells. However, the increased light exposure can lead to phototoxicity, necessitating a balance between image quality and cell health. Zeiss Campus, Magnet, FSU, Edu. 6. Spinning disc confocal microscopy. Spinning disc confocal microscopy utilizes a disc with multiple pinholes to scan the specimen rapidly, enabling high-speed imaging with reduced phototoxicity compared to traditional CLSM. This method is suitable for observing fast cellular processes in real time, such as vesicle trafficking and cytoskeletal dynamics. Abidi, Com. 7. Total internal reflection fluorescence TIRF microscopy. TIRF microscopy selectively illuminates and excites fluorophores in a thin region near the cell substrate interface, typically within 100 to 200 nanometers of the cover slip. This technique is ideal for studying events that are near the plasma membrane, such as receptor-ligand interactions and exocytosis, with high signal-to-noise ratio. ANATOMYPUBS, Online Library, Wiley, Com. 8. Multiphoton microscopy. Multiphoton microscopy employs longer wavelength light to excite fluorophores through the simultaneous absorption of two or more photons. This approach allows for deeper tissue penetration with reduced phototoxicity, making it suitable for imaging live cells within thick specimens or whole organisms. It is particularly advantageous for intravital imaging and studying cellular interactions in their native environment. Abidi, Com. 9. Light sheet fluorescence microscopy. Light sheet fluorescence microscopy illuminates the specimen with a thin sheet of light, reducing photo damage and enabling rapid, high-resolution imaging of live cells in 3 dimensions. This technique is beneficial for observing developmental processes and large-scale cellular dynamics over extended periods. Abidi, Com. 10. Considerations for live cell imaging. When selecting a microscopy technique for live cell imaging, several factors must be considered. Phototoxicity. Excessive light exposure can damage cells and alter their behavior. Employing techniques that minimize light intensity and exposure duration is essential to preserve cell viability. Temporal resolution. Capturing fast biological events requires imaging methods with high temporal resolution to accurately record dynamic processes. Spatial resolution. The level of detail needed dictates the choice of microscopy, with some techniques offering higher spatial resolution to visualize subcellular structures. Fluorophore selection. Choosing appropriate fluorescent probes with minimal photobleaching and phototoxicity is crucial for prolonged imaging sessions. Environmental control. Maintaining optimal conditions e.g., temperature, pH, humidity during imaging is vital to ensure physiological relevance and cell health. 11. Applications in zoology. Live cell imaging techniques are indispensable in various zoological research areas. Developmental biology. Monitoring embryonic development and cell differentiation in real time provides insights into growth patterns and morphogenesis. Neurobiology. Observing neuronal activity and synaptic interactions enhances understanding of nervous system function and behavior. Cellular physiology. Investigating intracellular processes, such as signal transduction and organelle dynamics, elucidates mechanisms underlying cell function and adaptation. Pathology. Studying pathogen-host interactions and disease progression at the cellular level aids in identifying therapeutic targets and strategies. 12. Solved previous year question. Question. Discuss the advantages and limitations of total internal reflection fluorescence TIRF microscopy in studying membrane-associated processes in live cells. Answer. TIRF microscopy offers several advantages for studying membrane-associated. Sources. You said, phase contrast. ChatGPT said, phase contrast microscopy. Phase contrast microscopy is a powerful technique for visualizing live, unstained cells. It enhances the contrast of transparent specimens by converting phase differences in light passing through a sample into intensity variations. This method is widely used in zoology for studying cell morphology, motility, and internal structures without the need for staining or fixation, which can alter cell physiology. 1. Principle of phase contrast microscopy. Light waves passing through a specimen experience phase shifts due to differences in refractive indices between cellular components and the surrounding medium. These phase differences are typically invisible to the human eye. Phase contrast microscopy transforms them into differences in brightness, making transparent structures distinguishable. A phase annulus in the condenser focuses hollow cone-shaped light onto the specimen. Light passing through the specimen is either undisturbed background light or delayed by different refractive index regions, diffracted light. A phase plate in the objective lens selectively retards or advances the phase of diffracted light. Interference between background and diffracted light enhances contrast, creating a bright or dark image depending on phase shifts. This method effectively highlights internal structures like nuclei, mitochondria, and vacuoles, making it ideal for observing living cells in their natural state. 2. Advantages of phase contrast microscopy. Visualization of unstained specimens. Unlike bright field microscopy, phase contrast allows detailed observation without staining, which is useful for studying living cells without altering their physiology. Enhanced contrast. Cellular components such as organelles, cytoplasmic granules, and membranes become visible due to increased contrast. Non-invasive technique. Cells remain viable, making phase contrast microscopy ideal for time-lapse studies and live cell imaging. Easy to use. No complex sample preparation is required, making it a quick and effective technique for biological research. 3. Limitations of phase contrast microscopy. Halo artifacts. A common issue where bright halos appear around specimens, reducing image clarity. Not suitable for thick specimens. Phase contrast works best with thin, transparent samples. Thick specimens can create overlapping phase shifts, leading to poor image quality. Limited color information. Since phase contrast relies on differences in refractive index rather than staining, it does not provide natural color contrast. 4. Applications in zoology. Cell culture studies, observing live cell behavior, growth, and division in tissue culture. Microbial and protozoan research, studying motility and morphology of bacteria, protozoa, and microscopic aquatic organisms. Sperm motility analysis, assessing sperm viability and movement in reproductive biology. Neuroscience research, observing live neurons and glial cells to study cellular interactions in real time. Parasitology, examining live parasites and host-parasite interactions in unstained preparations. 5. Solve previous year question. Question. Explain the principle of phase contrast microscopy and discuss its advantages and limitations in studying live cells. Answer. Phase contrast microscopy exploits differences in the refractive indices of cellular structures to generate contrast. It uses a phase annulus in the condenser to direct light as a hollow cone onto the specimen. Light that passes through cellular components undergoes phase shifts, which are selectively altered by a phase plate in the objective lens. Interference between background and diffracted light enhances contrast, allowing visualization of transparent, unstained structures. Advantages include non-invasive imaging of live cells without staining, enhanced contrast for internal cellular components, simple and rapid observation of biological samples. Limitations include the presence of halo artifacts that may distort image clarity, ineffectiveness for thick specimens due to overlapping phase shifts, lack of color contrast, requiring alternative methods for specific molecular identification. Overall, phase contrast microscopy is a valuable tool in zoological research, particularly for observing live cells in their native state. 6. Multiple choice questions MCQs 1. What is the primary advantage of phase contrast microscopy? A. High magnification power. B. Ability to observe live cells without staining. C. Three-dimensional imaging of specimens. C. Enhanced color contrast in specimens. Answer. B. Ability to observe live cells without staining. 2. Which component of a phase contrast microscope was responsible for creating phase shifts in light? A. Condenser lens. B. Phase plate. C. Objective lens. D. Eyepiece. Answer. B. Phase plate. 3. What is a major limitation of phase contrast microscopy? A. Requires staining for contrast. B. Only works for thick specimens. C. Produces halo artifacts around specimens. D. Cannot be used for live cell imaging. Answer. C. Produces halo artifacts around specimens. Phase contrast microscopy remains an essential technique in zoology, particularly for studying dynamic cellular processes without disrupting biological integrity. You said. Fluorescent and confocal microscopy. ChatGPT said. Fluorescent and confocal microscopy. Fluorescent and confocal microscopy are advanced optical techniques widely used in zoology and life sciences for visualizing specific biomolecules, organelles, and cellular processes with high precision. These methods utilize fluorescence to achieve enhanced contrast and resolution, allowing detailed examination of both fixed and live specimens. 1. Fluorescence microscopy. Principle of fluorescence microscopy. Fluorescence microscopy relies on the phenomenon of fluorescence, where certain molecules fluorophores absorb light at a specific excitation wavelength and emit light at a longer emission wavelength. This allows selective visualization of cellular components tagged with fluorescent dyes or proteins. A light source, e.g., mercury, xenon lamp, or LED, emits excitation light of a specific wavelength. A dichroic mirror reflects excitation light toward the sample but allows emitted fluorescence to pass through to the detector. A fluorescent dye, e.g., DAPI for DNA, FITC for proteins, binds to specific cell structures and fluoresces upon excitation. The emission filter ensures only fluorescence from a sample reaches the detector, improving contrast. Types of fluorescence microscopy. A. Epifluorescence microscopy. The most common fluorescence microscopy technique, where the entire specimen is illuminated and emitted light is detected. Suitable for fixed cells and some live cell imaging. B. Total internal reflection fluorescence, TIRF microscopy. Selectively illuminates a thin section of the sample near the cover slip, approximately 100 to 200 nm, minimizing background noise. Ideal for studying membrane-associated events like receptor-ligand interactions. C. Super-resolution microscopy. Techniques like STOR, stochastic optical reconstruction microscopy, and POM, photoactivated localization microscopy, surpass the diffraction limit, achieving nanometer scale resolution. Used for high-precision molecular and cellular studies. Applications of fluorescence microscopy in zoology. Localization of proteins. Fluorescently tagged antibodies help visualize protein distribution in cells. Live cell imaging. GFP, green fluorescent protein, allows real-time tracking of cellular events. Pathogen identification, used in microbiology to detect bacteria, viruses, and parasites. Neurobiology helps study neural networks, synapses, and brain activity in live tissues. Limitations of fluorescence microscopy. Photobleaching, continuous exposure to excitation light can degrade fluorophores. Phototoxicity, high-intensity light may damage live cells.